So in this video, we are going to go over the replication fork and the enzymes, the most important enzymes that are involved in microbial DNA replication. So there are 5 different enzymes. The first is helicase, and helicase is responsible for unwinding the DNA helix. And it helps to open up the DNA and make it prepared to be copied. We then have Primates. Primates is an enzyme that builds a small primer that is needed in order to start DNA replication, and I'll talk about that again here in a second. And the primer is made up of RNA. All right, the next is DNA polymerase 3. And DNA polymerase 3 is the enzyme that is actually building the new sequence of DNA. So it builds the new sequence, or the copy, by putting together a polymer of DNA. So it puts together nucleotides. And then DNA polymerase 1, this comes back and it fills in holes that are in the lagging strand, and I'll talk about that again when I actually draw the fork and how that arises. And it also does a little bit of proofreading. DNA polymerase 3 also does a little bit of proofreading just to make sure we're putting the right bases in the right positions. And then finally we have ligase, which really just kind of wraps it all up. It helps to seal the spaces in between the Okazaki fragments, so it seals the lagging fragments, and it also helps to unwind or completely separate the two daughter chromosomes. So separates daughter chromosomes at the very end. Right, so these are the enzymes that you need to know. There's more involved than this, really, but these are the 5 most important ones. So let's go ahead then and look at the replication fork. So remember that microbes have one circular chromosome, and it's double-stranded and it's helical. However, helicase, we're going to imagine that helicase has already come in and unwinded the helix and opened up the chromosome so that we can see how replication is going to occur. So I'll draw the replication fork, and again, this is what I told you that I'm going to expect you to be able to draw and label for your test. Okay, so we have the 2 different sides of the circular chromosome here. This is the interior side, and this is the exterior, and we've seen it open up right here. So that's where the helicase has come in, it's unwound it, and it's opened it up. Now, the other thing that has happened is the primer has already been built. So remember that a primer is a short sequence of nucleotides that is going to complementary base pair to some position, some starting position on the DNA. And the reason why it is necessary is because DNA polymerase cannot just start building a polymer of DNA. It can only attach DNA to a sequence that already exists. So that's why we have to make the primer that will kind of hook on to the DNA, and it provides a position for the DNA polymerase 3 to come in and start adding nucleotides and building a copy of our original chromosome. So there's enough base pairs there that it's attracted here just by hydrogen bonding, because there's enough A, T, C's, and G's that are matching up, or complementary base pairing to here. Excuse me, not T's, U's, because that's RNA. All right, so the other thing that's important about this chromosome is to look at the directionality of the DNA. So in the last video, I talked about the structure of DNA and I explained the directionality. So it is 3' to 5' on one side, and then on the other side, it would be 5' to 3'. So it's an anti-parallel arrangement. So as DNA polymerase comes in, it's going to start adding nucleotides, and we actually have 2 big DNA polymerases that start here and they work their way out. So I'm going to kind of draw them here as if they had started in the middle and moved to this edge and then would continue along down this path, right? Now, the problem with DNA polymerase is it can only build DNA in one direction. It can only go 5' to 3'. It can't go 3' to 5'. So let's sort of figure out if it's moving in this direction. This would be we would have 3' here to 5'. So that means that the direction of the other side would be 5' to 3', right? So that's the right direction. So the polymerase would have been building along this way in a nice, smooth, just adding a nucleotide, adding a nucleotide, adding a nucleotide, and it has this nice, smooth-looking strand, okay? This strand we call the leading strand. Now, when the DNA polymerase is moving along, it actually is attached to both sides of the DNA, so it's not only creating a leading strand up on this side, but it's also adding nucleotides here. However, remember that this one would now be the wrong direction. So this has 5' to 3'. So up here, the new sequence that we would be building would be in the 3' to 5' direction, which is the wrong way. So what DNA polymerase has to do is it has to scoot forward and code backwards, and then scoot forward and code backwards, and scoot forward and code backwards. And as a result, we end up with these little fragments which make up what is called the lagging strand. And as a result of discovering these, the guy who discovered them got them named after him, so they are called Okazaki fragments. Now, likewise, this DNA polymerase in the other direction is going this way. So if we look at the directionality there, here is 5' and 3' is over there. So in this way, we would have 5' here and 3' here to match up to the other side of the DNA. And so as this guy is going along, his lagging strand is actually going to be up here because he's going the wrong way in this direction up top. So his fragments would be like this. Whereas down here, he would be going in the correct direction. So the leading strand as a result of him would be here, right? So they make their way all the way around until we have two brand new daughter chromosomes, one of which is the old strand, and one of which is the new strand. So half of it would be leading and the other half would be sort of lagging like this. And then you've got your other daughter chromosome, and it has some lagging and some leading. And then in comes DNA polymerase 1, and it checks to make sure everything's okay. And then ligase comes in and seals up and solidifies that lagging side and helps to completely separate them apart. All right? So that is DNA replication for microbes. And what I will be expecting you to draw and label on the test will be what you see here, including labeling the primer and labeling the DNA polymerase enzymes.